They are separated, the two single DNA strands can act as templates for the production of two new complementary DNA strands. Remember that the double helix consists of two anti-parallel DNA strands with complementary 5' to 3' strands running in opposite directions. Polymerase enzymes can synthesize nucleic acid strands only in the 5' to 3' direction, hooking the 5' phosphate group of an incoming nucleotide onto the 3' hydroxyl group at the end of the growing nucleic acid chain. Because the chain grows by extension off the 3' hydroxyl group, strand synthesis is said to proceed in a 5' to 3' direction. Even when the strands are separated, however, DNA polymerase cannot simply begin copying the DNA. DNA polymerase can only extend a nucleic acid chain, but cannot start one from scratch. To give the DNA polymerase a place to start, an RNA polymerase called primase first copies a short stretch of the DNA strand. This creates a complementary RNA segment up to 60 nucleotides long that is called a primer. Now DNA polymerase can copy the DNA strand. The DNA polymerase starts at the 3' end of the RNA primer and, using the original DNA strand as a guide, begins to synthesize a new complementary DNA strand. Two polymerase enzymes are required, one for each parental DNA strand. Due to the anti-parallel nature of the DNA strands, however, the polymerase enzymes on the two strands start to move in opposite directions. One polymerase can remain on its DNA template and copy the DNA in one continuous strand. However, the other polymerase can only copy a short stretch of DNA before it runs into the primer of the previously sequenced fragment. It is therefore forced to repeatedly release the DNA strand and slide further upstream to begin extension from another RNA primer. The sliding clamp helps hold this DNA polymerase onto the DNA as the DNA moves through the replication machinery. The sliding clamp makes the polymerase precessive. The continuously synthesized strand is known as the leading strand, while the strand that is synthesized in short pieces is known as the lagging strand. The short stretches of DNA that make up the lagging strand are known as Okazaki fragments. Before the lagging strand DNA exits the replication factory, its RNA primers must be removed and the Okazaki fragments must be joined together to create a continuous DNA strand. The first step is the removal of the RNA primer. RNase H, which recognizes RNA-DNA hybrid helices, degrades the RNA by hydrolyzing its phosphodiester bonds. Next, the sequence gap created by RNase H is then filled in by DNA polymerase, which extends the 3' end of the neighboring Okazaki fragment. Finally, the Okazaki fragments are joined together by DNA ligase, which hooks together the 3' end of one fragment to the 5' phosphate group of the neighboring fragment in an ATP or NAD dependent reaction. We are now ready to review the steps of DNA replication. The process begins when the helicase enzyme unwinds the double helix to expose two single DNA strands and create two replication forks. DNA replication takes place simultaneously at each fork. The mechanism of replication is identical at each fork. Remember that the proteins involved in replication are clustered together and anchored in the cell. Thus, the replication proteins do not travel down the length of the DNA. Instead, the DNA helix is fed through a stationary replication factory, like film is fed through a projector. Single-strand binding proteins, or SSBs, coat the single DNA strands to prevent them from snapping back together. SSBs are easily displaced by DNA polymerase. The primase enzyme uses the original DNA sequence as a template to synthesize a short RNA primer. Primers are necessary